Confirmed COVID-19 cases continue to grow around the world. But why do only a small percentage of people become seriously ill? With genomics, we can detect, diagnose, and track the virus. But how do we predict who gets sick and who is resistant? Precision medicine aims to understand how disease impacts an individual from diagnosis to treatment, instead of the one-size-fits-all approach in traditional medicine. Essential to this research is proteomics, which is the study of all proteins within an individual. One of the Proteomics partner labs is the Blackburn Group Core Proteomics facility, which is found within the Institute of Infectious Disease and Molecular Medicine at the University of Cape Town. The lab uses state-of-the-art proteomic methods and instrumentation to develop diagnostic tools to better understand how diseases progress and how people respond to treatments. Some good examples of the group's output include the development of point-of-care tuberculosis tests using urine samples, or the adaptation of that technology to the COVID-19 space to help better understand and predict disease trajectory of individual patients. Professor Jonathan Blackburn, principal investigator for the group, is passionate about the translation of proteomic research into tangible outputs that benefit society. He is excited about the continued growth and application of proteomics within mainstream biology and the biomedical space.
So the classic proteomic technologies revolve around use of extraordinarily expensive mass spectrometry instrument. Now, mass spectrometers you can think of as a giant and extremely expensive weighing scale. So we measure the masses of molecules and we quantify the amount of molecules, and from the weight of the molecule we can usually identify individual proteins within a sample. And then we look for dynamic changes in that abundance that correlate with disease. So one of the things we've started to see recently in proteomics, both using our protein array technologies and also using the mass spectrometry methodologies that my group use, is that if you take a cohort of patients who've all been characterized as having the same disease, let's take colorectal cancer as an example, that actually when we look at the proteomic level, those individuals are not all the same, and actually they, at the protein level and the proteomic level, they seem to fall into discrete subtypes of disease. So one of the things that's interesting in the COVID-19 space is that PCR-based tests might tell you that you've got the virus, but it won't tell you what's going to happen next. And so we're very interested to understand whether the changes that happen in your body in response to infection might manifest in terms of a change in the abundance of proteins that we can measure. And we think that the technological approaches we've developed will give us insight that other approaches have not been able to do to date. And so where I see proteomics going is that we need increasingly to set up core facilities, and this is one of the jobs of Diplomics, that will enable the technologies that we've developed and the expertise that we've brought in to become available to a much wider repertoire of research groups at UCT and through the wider community in South Africa.
For more information, please contact the Blackburn Group using the information displayed or visit the Diplomics website.